Hi everyone, let's start with some further concepts of genetics. Today we will be doing something very interesting. They are human genetic disorders. So humans can have somatic disorders, which are, let's say, most disorders that we have, like infectious diseases, right? So infectious diseases are not really genetic in nature. They happen to us because we have been affected by some kind of a bacteria, virus, or a protozoa or a fungi, which has given us an infection. And these are infectious diseases which can be communicated. However, there is a vast group of diseases in humans which are caused due to multiple reasons. And their most common reasons are either mutations to genes, somatic or X-linked genes, or chromosomal abnormalities. Some of these disorders, because they inherit with a Mendelian pattern, are known as Mendelian disorders. And some of them, which happen due to some kind of a malfunction in the cell during meiosis, so we call it disjunction of chromosomes, where, see, during meiosis the 46 or let's say 44 plus XX of the mother and the 44 and the XY of the father breaks such that you get 22 plus X, 22 plus X on one side, that is in the females, and in males you get 22 plus X and 22 plus Y. However, sometimes due to non-disjunction, so this is disjunction of chromosomes. If non-disjunction were to happen, then instead of breaking exactly into a haploid number of 23, what happens is that, let's say 23 plus X comes here. Okay, so one whole set comes here. As a result, you would have 21 plus X in this group. So let's say chromosome number 21 failed to break its homologous pair into two different sets. Then this organism or this ova, when it will fertilize, it will give you two chromosome 21s, that is the whole homologous pair already from the mother, and one will come from the father. So the organism, the zygote, will end up having 3 copies of the chromosome 21, right? So that is non-disjunction. So in very simple words, however, we will be doing this whole thing in quite a lot of detail. So non-disjunction means no splitting or non-splits of homologous chromosomes. Okay, that gives rise to unequal numbered chromosomes in different organisms, and therefore are known as chromosomal abnormalities or aberrations. Okay, for now, for this video, we are going to focus mostly on understanding how Mendelian disorders work, and this will come out as a series of videos, right? So Mendelian disorders are called so because they have a Mendelian method of inheritance. So they have a dominant recessive mode of inheritance. So for all of these diseases, which you will learn how to analyze, how to understand from a pedigree, you will always consider these diseases to be associated with a single gene which has 2 alleles, one being dominant, one being recessive. Now, depending on whether the gene is autosomal or sex-linked or present in the mitochondrial DNA, you have 3 kinds of Mendelian disorders, right? So you have autosomal Mendelian disorders. So autosomal Mendelian disorders are disorders of the somatic chromosome or somatic chromosome genes. So the 22 chromosomes that we have, 22 pairs that we have, the first 22 pairs of chromosomes, those are your somatic chromosomes. Those are the chromosomes which give you your somatic features, right? Features of the soma or the body. So then those if there is a mutation in any of those genes, we would call that an autosomal disorder, right? Now, if the mutation is of dominant kind, then it becomes an autosomal dominant disorder, right? However, think about this. If there is already an autosomal dominant disorder in an organism, so the organism is born with a disease. I mean, not organism, let's say human in this case. If he or she is born with a disease, it's almost unlikely that this disease would be passed on to the next generation, because the disease can entail infertility, or the disease can lead to a negative selection in the society. Correct? So anybody with a disease, with a dominant disease, usually does not survive to fertility. And if that happens, then the disease would gradually decrease in the population, right? Because they are not reproducing, they are not passing on their dominant alleles which are mutated or which are faulty to the next generation, and therefore the disease will decrease in numbers. The number of people, the incidence of the disease, the frequency of the disease will decrease in the population. However, there are a few diseases which have a very interesting way around this. These diseases, like Huntington's chorea or Alzheimer's, they also have huge genetic components to it. Alzheimer's leads to plaque buildup in the brain, which leads to severe inability in retention of memory and recollection of memory as well. In Huntington's chorea, there is a kind of a muscular dystrophy that happens and leads to multiple kinds of movement failures, and eventually it's fatal. But both these diseases have a way around this phenomenon, where this dominant disease is expressed at or after reproductive maturity. So somebody with Huntington's chorea would not know that they have Huntington's chorea or Alzheimer's till they are well beyond their reproductively fertile ages, right? So which means that it will express itself at the age of 45, 50, 60, old ages, right? And therefore, as a result, these organisms would have well passed on their genes to the next generation because they thought they were healthy, and the disease stays in the population. However, certain diseases which are lethal in nature and are dominant as well tend to not get passed on to the next generation, and this kind of pressure is known as a negative selection pressure. So because it is a character which eventually leads to a disability or inability to adapt to the environment or to survive, therefore it gets negatively selected as per Darwin's theory of positive or natural selection, survival of the fittest. Not the fittest, therefore negatively selected, right? Moving on to recessive disorders. So all of these 22 chromosomes, 22 pairs of chromosomes, if the genetic mutation is recessive in nature, so that means that in the presence of the homologous chromosome, right, if one is not diseased, this is, let's say, chromosome number 21. If this is a normal chromosome and this is a diseased chromosome, the individual would be normal because the mutation is recessive in nature. However, only if both of them are mutated, both the chromosomal alleles are mutated, then you have the condition of a disease. So it's a very similar concept to Mendelian genetics, where a homozygous or a homozygous dominant or a heterozygous condition gives you the dominant feature. So for dominant diseases, homozygous dominant and heterozygous dominant will give you disease. in this case will not give you the disease, right? There are multiple diseases which come under these recessive somatic disorders. So for example, alkaptonuria, phenylketonuria, both of them are metabolic diseases. Albinism is a disease where melanin does not get formed. It's a skin color disorder. Thalassemia is a blood disorder where either alpha or beta hemoglobin is not formed properly. Excuse me. There are multiple others like Tay-Sachs disease, Caucher's disease, which we will do in detail because you have it in your syllabus. Excuse me. Moving on to sex-linked disorders. So the last set of chromosomes, that is your X chromosome or your Y chromosome, if the aberration or the mutation is in the X or the Y, you have X-linked or Y-linked diseases. Now, X-linked diseases can be either recessive or dominant, right? Because in females, again, you would remember that there are two X's, correct? So, X-linked diseases can have a dominant or a recessive mutation. However, for Y-linked diseases, try and understand this very well. Only males carry Y. Am I right? So if a father is diseased, only his sons can get that disease from the father, not the daughters, because daughters will never have Y. So if there is a mutation in a Y chromosome, females are never predisposed to Y chromosome diseases. Why? Because again, we don't have Y chromosomes, whereas males have a Y chromosome. Therefore, if their fathers have the disease, then the disease will always come to them because the father will always contribute his Y. If this Y is diseased, usually show diseased with a dashed, the daughters would never get it. But all sons will always get it because this Y will always come to the sons from the father, right? So Y-linked disorders are also basically paternally inherited male disorders. I hope this is clear, right? Now moving on to mitochondrial diseases. As we know that mitochondria and chloroplasts are the two of the autonomous, semi-autonomous organelles that we talk about, right? Why are they semi-autonomous? Because they contain their own DNA. If there is a mutation within the genes of those DNA, then we can have a disease which is known as a mitochondrially inherited disease. Now, the interesting factor about mitochondrially inherited diseases is that when fertilization happens, the sperm contributes only its haploid nucleus and the proximal centriole to the ovum. Inside the ovum, all cell organelles are already present. Right? When the sperm's nucleus enters, the pronuclei of the female and the pronucleus of the male, they fuse and a zygote or a hapdiploid nucleus is formed. That is your zygote, which basically means that all organelles that are present in the single-celled zygote is maternally derived, right? The father does not contribute to any of those organelles. Therefore, all our mitochondria that I have in my body, I've gotten all of them from my mother and not a single one from my father, right? So, if a mother has a mutation in a mitochondrial gene in her body, then all her kids will always get that disease. However, from the mother, let's say the mother has a disease, she has a son and a daughter. The disease will be passed on to both of them. However, if the daughter further has sons, let's say, then they will get the disease. The disease will pass because it can only pass from mother to son. But the son will never have the disease. The son's kids, if she is a daughter, let's say, she will never have the disease. Do you understand why? That's because the mitochondria is only inherited from the mother, right? From the females. So the mother will give to the daughter and the daughter will pass it on, right? Now, if the daughter has only sons and no daughters further down, then again the disease will stop here. It will not go further down. However, this son, unless and until the wife also has the same disorder, his family from here down will never have the disease again. Correct? So that is the interesting part of mitochondrial disorder. It actually tracks back to the concept of the mitochondrial mother, and therefore they are maternally inherited disorders. There are multiple disorders, but they are very uncommon. They are rare disorders, so you do not have it in the scope of your syllabus, and we will not discuss it. Just for the sake of interest, there is something called myeloid epilepsy, right? It's a kind of an epilepsy which happens. There is a stroke syndrome that happens. So multiple very rare but very complicated disorders happen, diseases happen due to mitochondrial inheritance. Now, all of these disorders, which you have to also understand each disease in detail, as well as how to identify them in a family. To identify a Mendelian disorder within a family, we do something which is known as a pedigree analysis. So to understand pedigree analysis, which we will start from next video, let's in this video understand the symbols, how a pedigree works, and so that you have some time to go through the ideas and the concepts, and we'll come back with the complete analysis for every disorder, right? So symbols are squares for males, circles for female. This is universal. Now, if the square or the circle is hashed in any way or solid, okay, so it can also be like this, solid. Usually in prints you get solids. Then it means it's a diseased human, okay, diseased male, diseased female. If there is a dot in the center, that means it's a carrier. However, carrier, the concept of a carrier is something that you will understand, and it is one of the most important things to identify in a pedigree. So in most cases you will not get questions where the carrier is marked, but still for boards it's important to understand, because sometimes they give you the symbol and ask you what it means. So a dot in the center means the carrier, okay. A single line between a male and a female means a marriage. A line coming down vertically from that single line means the line of child or the line of bearing, correct? So from there, if they have a single child, it would come out and be like this. If they have two children or more, then you would branch it out, okay? Now, if there is a double line between a male and a female within the same family, so you can see that they are siblings, right? So these are the parents, they have two kids, one daughter, one son, and they got married. That is known as a consanguineous marriage. In multiple sects of the society, in India and abroad, this is a very common norm. However, as we know that inbreeding leads to a certain kind of depression. So let's say there is a disease that runs in any of these families, in this family, right? So there's a good chance that both of them have the disease or carry the disease. Therefore, the moment there is marriage within the same family or very closely related humans, there is a chance of passing on these genetic disorders furthermore. However, it's a very common trait and it's known as a consanguineous marriage. Questions like, you will be given a pedigree to analyze where there are a lot of consanguineous marriage and a lot of occurrence of a certain disease. Questions like, why do you think this family has occurrences of this disease, has such occurrences of this disease? Why do you think this disease happens to so many people in the family? In that case, consanguineous marriage can be named as a reason behind the multiple occurrences of the same disease within a family. Now let's look at one pedigree. So this is a pedigree, right? It's a random pedigree. Nobody in this pedigree is diseased. Everybody is disease free. So this is the parental generation. We call them, or we name them with Romans. So it's Roman 1, individual 1 and individual 2. They have a total of 2 kids, 1 daughter and 1 son. So that's generation 2, individual 1. Now the daughter, which is individual 1, gets married to a man from outside, and the son gets married to a woman from outside, and they have their kids. So their kids become generation 3. In generation 3, you have the first couple, the daughter has 2 daughters and a son. The second daughter and the third, the son, right? The individual 2 and 3, they have a consanguineous marriage. In the second family, so this is family 1, this is family 2 now. This was first family. In the second generation, there are 2 families. In the third generation, there are 2 families. Here, this family has 1 daughter, and I think they have two daughters, right? So they have 2 daughters. The first daughter gets married, the second daughter is unmarried. The first daughter has a daughter again. So this is generation 4. So from the consanguineous marriage, you have you have a daughter who gets married to somebody from outside, and from the second family, you have 1 daughter. In generation 5, you have 2 daughters. So what we do is that we name each generation with Romans, and each individual does not matter in what order they were born or in what order they got married, just in the order in which they are present in the pedigree, as this. Now there is one more symbol, which is, let's say, this. In this family, if there is an arrow on one organism or on one human, that means whatever disease runs in this family was first primarily reported by this individual. Okay, I hope this much is clear. We will continue with pedigree analysis for all of these kinds of diseases in detail in the upcoming videos. Stay tuned. Thank you.